Okay, hello there. I am here to talk about the various ways to measure infant perception or otherwise research it. To start off with, infants, they have a number of ways that we can sort of get an idea of how they're cued when they see something, right? It's difficult for them to be measured simply because they can't really answer or fill out questionnaires or do anything conventional, so we have to use context clues in the body language. The various tools that we have include stuff like naturalistic observation, which is just watching them, film and video analysis, which is the same thing but recorded, changes in heart rate and respiration, similar to the other somatic measurements you can take on an athlete or anybody else. There's also non-nutritive sucking, which is a very special type of measurement found by, let me see if I can get the source here, Sickland and DeLucia in 1969, which essentially entails the use of a pacifier, which is basically a scientific pacifier that counts and measures suckling patterns in order to see what the reflex is, see what they do with their mouths in response to stimuli. There's also habituation slash dishabituation and evoked potential, both of which are specialized ways of measuring responses and stimuli in infants. There are kind of both processes by which to do that, right? Evoked potential is similar to heart rate and respiration. It's a physiological sign. It's where electrodes are attached to the head and brain signals are measured in response to stimuli. But habituation slash dishabituation is more complicated. The best way I can explain it is that it is essentially the process of first getting a stimulus that the child reacts to and presenting it to them so that we can get a reliable idea of how they respond to that, and then giving them another stimulus and seeing if they react at all differently or if they do react, or if they do so, how it is different, right? Yeah, that's kind of another way we can do things. It's more clear than straight up seeing what they do the first time around. It gives a control group, so to speak, for stimulus response.